we'll talk about specifically what it means to be a dynamic university resource and some of the things we think about in that concept of rigor and transparency. So these are your speakers for today. So thank you. I don't need to say much about the introduction, but some of you may know me right from Biostat 521. Potentially, some of you are taking it in the fall. I'm right after I'm finished with this, I'll go back to prepping for that class. And so then Brett, who's also here. So thanks to Brett for joining. Brett spends all his time working on the precision health work and the Michigan Genomics Initiative. So happy to have him involved too. So to start here, I want to motivate, why we even have this data set, this Michigan Genomics Initiative. Well, it's actually part of a larger precision health initiative at the University of Michigan. And that's almost certainly a term that you've heard of, the precision health, but it means different things to different people. So for some people, it means discovery, and that means, finding new ideas, understanding better the way that the biological systems work. As people who are in, as a statistical geneticist and you are bioinformaticians, maybe this is the area that you think about the most in terms of discovery, but that's not it, right? There's also treatment, right? So how that you can take these discoveries and then apply them so that you can have personalized and targeted treatments of individuals who are sick, right? So finding the right treatment for the right person. And then there's also people who, when they think about precision health, they're thinking about avoiding disease and sickness entirely, right? And so how can we take these discoveries and put them into practice so that you don't even have to get to the treatment stage, right? So that people remain healthy, right? And so there's all these three ideas. So precision health is a big field, and there's lots of people working on it, including people like us who are quantitatively and computationally savvy. I'm a little rusty on my slide transitions apparently here. Okay. So regardless of whatever your research goal is with precision health, you need data and you need lots of it in order to get at that personalized, individualized level. Okay, so having lots of samples and lots of variables. So the University of Michigan Precision Health Initiative was started a few years back with support by the president, the provost, and deans of several schools here at the University of Michigan. And one of the major goals of precision health was to create these cutting edge data sets that could quickly push research at the university. So they use these words, which I like. They wanted to create data resources that were accelerators and incubators for interdisciplinary health research. And the idea was make these data sets available, okay, that could be used by many people for many purposes, and then don't put the onus on the researcher to create these data sets, okay? And so there could be many potential talks about precision health using the Michigan Genomics Initiative. They might focus more on individual scientific questions. And this talk, we're gonna focus more on the data, right? The data itself. How is this data resource created and how is it managed? And keeping an eye on the theme of rigor and transparency to enhance reproducibility. So the first step in having a good plan for a data resource like this is to get yourself an interdisciplinary team. And so Brett and I are just two members of a much larger team of people who work on this data set. So we fit over here or over here. I guess if you can see can you see my slides all right? Is that okay? Just want to make sure. The genetics team. This is housed within the biostatistics department. Then there's the clinical data, which we'll talk more about specifically, like different elements of data that fall into this. There's a whole unit that focuses just on the clinical data, doesn't touch the genetic data. The central biorepository, which is where the physical biospecimens are going to be stored. And then many different people who work within the precision health unit and some have academic appointments, they're professors, and some people are research staff members who are specifically working on these precision health things. So having this interdisciplinary data team where you have these complementary skill sets is hugely important, right? So we understand the genetic data really well, and then there's people who understand the clinical data much better than we do, but being able to come together and talk and bring our skills to this data set, right? And also, I just want to recognize that Brett and I don't do everything here. So what is the Michigan Genomics Initiative? Well, it was started in 2012 as part of a anesthesiology study of pain outcomes. That's when recruitment started. So it was started by a couple anesthesiologists, Chad Brummett and Sachin Ketterpal. And so they were collecting patients when they were coming in to U of M to have surgery done, okay? And so, this might seem kind of an odd time to try to corner people to recruit them to a study, but actually is kind of convenient because, among other things, if you are going to take a biosample from them, like a blood sample, you physically have to be with them, right? So this provided a nice opportunity to physically be with them, especially since the tube's already in them if they're preparing for surgery. It meant they were using the health system, right? So they had some clinical data available. And they were also present. They were able to go through the consent process and potentially also fill out surveys in advance. It's a longitudinal study, okay? So meaning that we have data collected at over time, right? And you'll see that's a function of having electronic health record data. And what happens is people consent to the linkage of their clinical health data to their biospecimen for research purposes. And actually, these people, when they consent to the Michigan Genomics Initiative, actually consent to being recontacted potentially, so that if investigators have follow-up questions, they can ask those questions as well. So I actually include this picture right here because it's kind of an interesting looking picture, but this actually comes from the consent form from MGI. Quite a bit of effort was put into creating something like this, a pictorial version of the consent, to try to explain to people. If you've never seen a consent form, they're often very text heavy, very hard to get through. Tough for people who are not researchers, tough for researchers even to get through sometimes, right? So this provided, this was sort of an experimental way to try to see if, the consent process could be made more understandable to the people that you were recruiting and approaching. And the returns seem to be pretty well that people understand it. So I like that picture. So where does data come from? So we've got two major sources of data that we're going to be considering today, and that's by no means all of it. First, we can think about the genetic data. So a blood sample is taken during the consent process. That blood sample is then taken to the Central Biorepository, CBR, which is located within the medical campus of the University of Michigan. It's the actual blood sample, a small amount is taken and sent to the U of M sequencing core, okay? But then the rest of the blood sample is biobanked in the CBR, okay? And it's documented, and it's actually accessible for future research purpose, right? And so, as we'll see, what we do is we extract genetic information, genotypes, but there's other things you can get from blood, right? And so somebody can go back into this and they can try to get expression data from this, right? Or they could go back and try to get a different type of DNA sample, so they can maybe do a sequencing study or something, okay? But that exists, and so this is the idea of a biobank, is that some physical biospecimen still remains. So once the U of M sequencing core receives that blood sample, they genotype using whatever array we're currently using. So right on this slide we have human exome core, and we've got a star there because we've changed it since. But there's genotype for about 500,000 SNPs on these arrays, and they do some basic quality control. Once they've done a handful of these, they will send them over to our biostatistics team, which is located in the School of Public Health. There we do extensive genotype quality control on these samples. We perform genotype imputation, so we bump up that 500K SNPs all the way up to something like 50 million SNPs. And importantly, what we do is we provide access and then analytic support to other people at the university that want to use this data. So you're probably somewhat familiar as people in bioinformatics with genetic data. maybe you might be a little bit less familiar with clinical data, right? So the thing that makes MGI unique and very helpful, very useful as a research tool is that it's linked to patient electronic health records, and these are digitized versions of a patient's medical chart. So I think it's potentially easy to lose sight of the fact that, you know, patient medical charts were not always digitized, right? You know, there was a time when you'd go to the doctor and there would be filing cabinets, right, of paperwork, right? And, you know, the doctor didn't enter things, or whoever was seeing you didn't enter things in on a computer. They wrote them down on pieces of paper. Those pieces of paper got filed into a folder. Those folders went back into the filing cabinet. That filing cabinet got closed. And that data, you know, nobody was using it, right? It wasn't data as we think about it, right? It was just a historical record, and it might not be looked at again until the next time you went to a doctor's office. Well, there was a movement towards digitizing these records. Okay, and then once you've got it digitized, that changed a lot, right? Because now you've got it in a form where you can easily query people's electronic health record data. Okay. What's important to know about electronic health records, though, is there's something called administrative data. They store things like diagnosis, procedures, lab results, lab test results, prescriptions, even some notes, right? Some free notes. But they're stored primarily for billing purposes and record keeping, right? So record keeping on a patient, and then billing purposes, right, to get back to insurance, to make sure people get paid, often used for analysis of hospital resources and quality metrics, right? So how well is a hospital performing at specific procedures? How successful are they? They are a historical record of treatment for a patient, but they're not, that data is not being taken primarily for research, right? And so there are some, you know, we could spend a lot of time talking about the pros and cons of EHRs for research, but again, the key is that now that it's digitized, you can easily query it, okay, and you can find out, you know, for example, you know, who in your data set, you know, has hyperlipidemia by searching for specific codes, right? So here's an example of an ICD-10 code, and these are sort of ubiquitous coding scheme that's used across the world, really, right? And so health systems use this. So it's a way of being able to easily query data. So we've got genetic data linked now with medical phenotypes through the electronic health record. Undoubtedly, you've heard of the term big data. So I happen to think this must count for big data. As of August, so that's now 2021, we've got something like 84,000 samples who've consented to be part of the Michigan Genomics Initiative. So that's a pretty big sample size, right? On each one of these people, we've got something like 50 million genetic variants. We also have in the electronic health record, we have over 22 million medical encounters for these 84,000 patients, right? And so the number of medical encounters is going to vary dramatically, right, between people, right? That'll be based on health, right, and also health utilization of the healthcare system, right? Do you go to U of M for all your procedures, or do you go to other places too, right? Did you travel to U of M to get some kind of specialized procedure, and maybe you see a primary care physician outside of the Michigan network? Okay, so I think this counts as big data. It's a pretty big data set, and it's getting bigger. Okay, so people are here at U of M are thinking about ways to expand this idea of a phenotype and what a phenotype can be. So specifically, there are efforts out, something called the EpiQ survey, to find out things like family history, social and environmental risk factors that people have. Okay, and now one of the reasons people do this is because this is sort of a blind spot for EHRs, right? As we said, right, you know, an EHR is a historical treatment of a patient's record, right? You know, well, your family history, you know, isn't going to come into the billing process, right? And so that might not be included in the EHR that well, right? So this is trying to get at some of the areas that are not well captured by the electronic health record. And then there's also studies that are trying to get at behavioral and lifestyle choices made by people within the study. So for example, there's something called the MyPack study, which actually gave Apple Watches to participants. And so the deal was, hey, we give you an Apple Watch, but we need to be able to track, you know, what your lifestyle and behavior things are, right? And so again, just many more types of data that are being brought into the Michigan Genomics Initiative. as part of this precision health movement. So it's big data, right? And what's cool about it is that it opens up new doors for analysis, okay? Because what you have is you have a whole genome, right? You have DNA, you're able to measure the whole genome. And then you have complete phenome, right? So put complete in quotes, but that electronic health record, right, is giving you, the complete medical history of somebody, right? So you can tell what they are, what diseases they have and potentially don't have. And you have it on the same individuals, right? And so, if you're familiar with the genome-wide association study, what you would do is you'd go out and you'd get a bunch of cases, you get a bunch of controls for a specific disease, and then you would use something like a logistic regression, right, to be able to test whether individual variants in the genome are associated with the trait of interest, right? So here is what the GWAS plot looks like for type 2 diabetes and MGI, right? And so this in and of itself would have been a major, major achievement, right? To be able to run this analysis and get a Manhattan plot like this. Well, when you've got something like MGI and you've already got the genetic data measured, well, now I can go to another phenotype, right? Because I can go into the electronic health record and check, right? So it doesn't have to just stop at one trait like type 2 diabetes. You can keep measuring other traits, okay? So hypertensive chronic kidney disease, right? Maybe another well-studied trait. But, you can also study traits that maybe aren't well studied, right? That don't have a lot of, and there's not a lot of money put into studying them. Restless leg syndrome, is there a genetic basis to that? This next trait, which I found, I had no idea what it was, but it's actually a defective closure of the eyelids. Lagophthalmus. Maybe one of you probably knows how to pronounce that better than me, right? But there's lots of, you can look at virtually any trait, right? That's code that you can get electronic health or ICD coding for. Okay. And then what you can do is if you have specific traits of interest, you can do something called a phenome-wide association study. So now I can take, instead of a genome-wide association study, which takes one trait and looks at it across millions, potentially genetic variants, what you can do is take one variant and now look at it across many different traits. So here's an example of a SNP that was associated with skin cancer. And every one of these dots on this plot now represents a different trait. They're colored by different categories of diseases. But you can see that, this trait, has obviously some things that are clearly related to skin cancer, but then there's some other conditions that this SNP is related to, and this allows you to look for things like epistasis, right? And try to figure out, again, having this data set lets you ask different questions. So I called MGI a dynamic university resource when I started. And so what does that mean to be a dynamic university resource? Well, dynamic means changing, obviously, as opposed to static, which is not changing, right? So MGI is changing over time. So first, it begins with the idea that, the enrollment is ongoing for MGI. So we're still collecting samples. So this plot here, I'm showing you what recruitment looks like over time, starting in 2012. And it's obviously increasing. The rate at which samples were collected was increasing. Right before the pandemic started, we were getting about 1,000 samples a month. You can actually see where the plot flatlines, right, in 2020, right, you can see essentially where the pandemic is starting, and recruitment has started to pick back up, but it was stopped for a while. Additionally, there's other studies that have tacked on to that original anesthesiology study that are focusing on different kinds of phenotypes using as the recruitment mechanism. So the MEN study is at an endocrinology lab. MHB is mental health. Big Bird is sampling children. And then I mentioned that MyPack study, which is getting at mobile health technology. Oh, and then another important thing, the data set obviously reflects the community seeking help or seeking medical care at the University of Michigan. And so the university or Precision Health is now trying to increase the ethnic diversity in the data set by creating alliances with other places. So doing recruitment in places like Dearborn, in Detroit, and then they're actually trying to look out of state to find other places. So that's sort of a work in progress and plans for the future for MGI. So the data set is dynamic in that it's changing, right? And so you can think about how this thing's changing, right? So, here I'm showing you on the left and the right some sort of cartoon pictures for what the genetic data and what the electronic health record data might look like. So clearly, we're recruiting samples ongoing. So the number of rows that you have is always increasing, right? But we also have the number of columns are constantly increasing as well, right? Well, so the reason why is because when you think about genetic data, there's always new technologies that are coming around that allow us to type new additional different types of markers, right? And so one of the things Brett will talk about is that what our change in an imputation panel means, and how we're able to get at more markers, but even using different technology, right? So as I mentioned, you could sequence these samples instead of genotyping them. You could measure methylation on them. You could measure expression on them, right? So all different ways that you could increase this data set. And then on the electronic health record end, right, again, of course, the number of rows is increasing because we're getting more samples, but these electronic health records are updated daily, right? And so every time somebody comes to the health system and has a new encounter and they get a new diagnosis, new labs taken, that stuff is being added, right? So you can imagine the number of columns, right, is increasing. So this is an electronic health, this is a data set that's constantly changing. That's the dynamic piece. The university resource piece is that we have to share this data set with the university research community, right? So how do you go about sharing a data set with the research community when it's constantly changing, right? And how do you ensure that it's of good quality? And also protecting, an important piece of this is how do you protect all the participants that are in this data set? So that's what we'll be focusing on the rest of the way. So we'll be hitting on elements of data quality, data integration, and then access and distribution, okay, and thinking about the rigor and transparency, but specifically focusing on this idea of dynamic, meaning that the data is accumulating and changing over time, and that it's a university resource, that we want to get this data to people, but we don't want to sacrifice security or compromise the confidentiality of patients. So I'll take a quick question break if anybody has questions. Okay, no questions, totally fine. I know I'm standing between you and lunch break, so I understand that. So let's talk about the genetic data quality control. So we receive in the biostatistics, our biostatistics team, we receive genetic data from the sequencing core in waves, right? And so, that should make sense, right? Because if people are being recruited to the study, over time, they give their sample, right? We can't wait until recruitment stops to do all the DNA work, to do all the genotyping, right? So, the sequencing core genotype samples as they can, in part that depends on their availability, what's going on in the sequencing core, what other kinds of work they have. But then we receive samples in waves, right? And so what kind of QC procedures do we do to ensure that we're getting good data? Well, we use multiple layers of QC, and that's what we're going to try to help you see here. So the first level, which is what I'll be talking about, is a sample level QC pipeline, right? So this is each individual sample is looked at individually, and we're trying to identify low quality, suspicious, or contradictory genetic DNA or data, and also if it's poorly matched to the electronic health record. So what this allows us to do then is it allows us to recheck to see if, well, was this the stock DNA that was bad, or was it the actual sample that was plated that was bad? Okay. Or is our manifest bad? Is our linkage between the genetic data and the electronic health record bad? Okay. Once samples pass through that, they start to build up. What we do is we incorporate them into data freezes, right? So you should probably be familiar with the idea of a data freeze, right? Where you say, okay, we're going to stop right now. We're going to look at this set of data in itself, and this is what we'll work with, even though there's other data building up. So we'll talk about the QC we do there to ensure that data is looking good. And to date, we've had 4 MGI data freezes. So pictorially, what's happening in this sample level quality control is that, you can imagine that we get a group of samples with their genotype data that shows up to us. In this little picture here, there's 5 of them. They each go through individually, right, a pipeline to look at multiple different metrics. Okay, and what our goal is to arrive at a set of high quality genetic data that's properly linked to clinical data. And so what we might do is we might decide that, samples 1, 3, and 4, they look good. Sample 2, we think there's a problem with it, that this data is not good at all. Okay, we need to rethink if we should regenotype this person or if the DNA that was originally taken was bad. And then there might be other samples that we say, well, there's something not quite right here, but the data might be good. Let's send this back to CBR, send it back to the sequencing core. Let's ask some questions and let's figure out what the problem is, and we might be able to rescue this data. So we run a variety of different summary measures on the genetic data. But what's important is that each one is trying to identify some specific problem with the data or try to help us better understand the sample. So for example, things that we run are a contamination rate filter, right? So trying to think about if it looks like, basically it's asked the question, does this DNA sample look like it's from one person, or does it look like it's a mixture of DNA from multiple people? And so, briefly that uses population allele frequencies, right, to look at increased or higher rates of heterozygosity than you might expect, okay? And it's actually really neat because in some cases you can actually figure out which two samples were mixed and contaminated each other. We look at things like genotype call rate, right? So if genotype call rate is low, it's usually a sign that the genotyping assay itself did not work. Looking for sex chromosome abnormalities, right? So in part, this is based on, does the sex chromosome that we see match the self-reported sex or gender that shows up in the electronic health record? Do we see evidence of large CNVs, right? And so large CNVs can be evidence of degraded DNA, okay? They also could be real, right? So we're just trying to identify potential problems. We also do ancestry PCA projection, right? And try to find out what is the ancestry of this person, right? And we can actually compare that to self-reported ancestry. So here's a list of the sample level quality control metrics that we use. And so I've ordered them here. And so we have things like technical issues, like a withdrawn sample. So people can consent to the study. They can actually withdraw from the study too. And if they withdraw from the study, we have to remove their data. But there could be plating errors, right? So you can have an entire plate that, from the time the samples were put on the plate to the time they entered into the machine, right? Somehow it got flipped or turned. So all your samples on a plate are wrong and it's hard to believe anything. We can have low call rate, high contamination rates. Particularly, we're often looking for duplicates, right? So have we seen this genetic data before? We compare a sample to every other sample that exists in MGI. And in some cases, we're interested in, does the IDs match, right? Or do the IDs not match? We're also looking, as I said, gender mismatches between reported and inferred sex, large chromosomal variants, and unusual XY compositions, right? So again, some of these, what I want to emphasize is some of these things could be real genetic. The genetic data is perfectly good, right? An unusual XY composition, that happens, right? But it's a way of checking to see they are also possible if there are artifacts in the data. So these things, again, as I said, what we're trying to do is get at specific problems. So if there's some kind of technical issue, we can't use that data at all. It's probably nothing that we can use. So that's that gray one. The green ones are getting at this issue of, is this a potential plating problem? Can we retry genotyping this sample, or is the stock DNA itself a mess? And so we have to send that back. It's either degraded or it's got high contamination in the stock itself. And that's something that we can coordinate with the sequencing core on. Then when it comes to these duplicates, right, we have to figure out, why are we seeing this duplicated sample? Okay, so anybody have any ideas why you might see another person show up that has a different ID but has the same genotype data? I'll have one interactive question. Does anybody have any ideas?
Identical twins.
Oh, Peter, look at you. Yeah, that's one potential explanation, right? So somebody could have you could have identical twins, and they have different IDs naturally. Another thing that can happen is that there are multiple studies recruiting samples, right? And so sometimes a problem that we run into is that, a patient shows up, they've already been recruited to one study that goes into MGI. Well, they show up to another study that study's got a different name. They don't know that they've already consented and that their data has been handed over. So they get a new study ID. They give their DNA. That DNA is obviously the same as it was before, right? And so we need to be able to catch these. And then also we have this issue of if we send people back to the sequencing core, if we get their data back, okay, well, that's going to match. And so we have to figure out which one's the better version. Gender mismatch. So part of the reason we do this gender mismatch is it's the simplest sort of the lowest fruit way that we can check if somebody's genetic data is paired to their electronic health record data properly, right? But there is the realistic case that when we have people that are transgender, right, their genetic sex and their self-reported gender might not match, right? So again, we talk to the CBR and try to figure out what's going on with these samples, right? Is this something that can be explained, or is this a data linkage error? So this slide briefly gives some idea about what our filtering techniques or what our filtering numbers look like. So you can see the duplicated samples. That is the biggest thing that we're dealing with. So 1,400 of this group of 63,000 on our current data freeze. And you can see, we don't have major numbers on any of the other categories, and most 95% of our samples clear all these hurdles with no problem. And then, I don't know, Brett, what the number is, but we probably have some number. How many that fail this initial pass through actually can be salvaged would be kind of an interesting number to figure out.
I don't have off the top of my head.
Yeah. I'm just thinking it would be something we should look into. Christina, do you have a question?
Yeah, so I was wondering these numbers, because when you identify an issue, and this is a related question, do you go back to the samples that you have already collected and check for that issue in those samples? And then these numbers reflect the whole database issue, or as you collect new samples from there on? Because I know this is checked as the samples are coming in. And let's say today you figure out there is an issue and you update the filtering and the inclusion of samples and the check of the samples. But do you go back and check the samples that are already in the repository?
Like if we change the metrics, is that what you mean?
For instance, you realize that you can have a duplicated sample today, and you include that in your code that checks when a sample is taken in. Do you go back to the samples that have already been collected and check if there are duplicates among them?
Yeah. So it's always checking against the full list of all the people that have made it into MGI, right?
Got it. So when a new issue is discovered, you go back to the database and try to fix the issue for the whole database. Perfect. That was what I was wondering. Thanks.
Yeah, and I mean, one of the things obviously when you've got two samples that have been genotyped and it's clear they're the same DNA sample that's been genotyped, we try to pick the one that's of higher quality, right?
Right. That makes total sense. Thank you.
Yep, good question. So those are so what I just talked about were the sample level QC procedures, right? So that's the individual samples. They show up, and we say, okay, we've cleared these. We trust these. Now, what happens is those are starting to build up on the side, but what we've got is another data set, what we call an analytic data set or a data freeze. That's what we're sharing with people at any given time. And once enough samples build up, we want to incorporate those new samples into our next data freeze so that we can have a larger data set, right, a larger sample size for people at the university to use. So the issue here is that we are genotyping people using this ongoing enrollment. MGI samples are genotyped in waves, as I mentioned. And again, this is one of the big ideas in molecular biology data, right? And it's something you as bioinformaticians should be familiar with, is that conditions for molecular assays are changing over time, be it things like the actual chemistry of the bead pools, the versions of the chips, the completely different chips, or new technology used to measure the chips. These things are changing over time. They don't stay the same. And so if you are wanting to have the same quality of data, right, within these people, right? You need to be keeping an eye on how these things change over time. And so when you have this potential for batch effects, right? And so batch effects are just differences between sets of genotype samples within these waves that have nothing to do with biology, but are just totally based on the experimental conditions, all right? So we do quite a bit of work to try to identify and weed out the potential batch effects that occur during this genotyping process. So I'm going to let Brett take over now for describing the work that really he does on keeping track of potential batch effects.
Yeah, so I'm Brett, the data analyst that works on this, and I do the genotype QC that Matt described and the preparation of the data freezes. And so that's what I'm going to talk about here a little more in detail. And so as Matt mentioned, these data are essentially generated in waves. Eventually, we have to merge the data together, but we have to be vigilant of batch effects, which again are effects on the data that come from some sort of non-biological aspect of the experiment. And so one really good example of where there's potential for batch effects is just the fact that samples have been genotyped across different array versions. And so there's been to date 4 different arrays that MGI samples have been genotyped on. 3 different versions of the core exome array backbone make up the bulk of it. And then now we're starting to genotype new incoming MGI participants on global screening array, a completely different array backbone. So we have to be able to evaluate batch effects between these arrays. Next slide, please. So to do this evaluation of batch effects between arrays, we leverage this negative control concept. So do we see signal where none should occur? So all things equal, we would kind of expect that allele frequency of SNPs that are genotyped across these different array versions should not be different. So we can perform a synthetic GWAS where we look at CHIP as outcome and determine if the allele frequency of individual SNPs is significantly different between CHIPs. There should be a figure that pops in here to the this is the outcome of the GWAS. You can see that there's some collection of SNPs that are associated significantly with one chip or another. So we have to go through and prune out these SNPs that have significant different allele frequency between the chips before merging the data gathered across different arrays together to perform the rest of the data freeze preparation. So next slide, please. So following merging data gathered across the different arrays, we can then use that merged data as input for the next steps in the pipeline, which are computational phasing and then genotype imputation. So phasing essentially tries to infer what the maternal and paternal haplotype is for each MGI participant, and then that phase data is then used as input for genotype imputation, which uses that phase and haplotypes from our reference panel to try and impute or estimate unknown genotypes that aren't directly assayed on the arrays themselves. This allows investigators to, for example, an association test association of millions of variants, most of which are imputed, versus the several 100,000 that are directly assayed on the chip itself. And I think the important thing to understand from this slide is that both these procedures are statistical in nature, and so they're subject to error. So we want to have a way to evaluate the error associated with these phasing imputation processes. Next slide, please. So phasing, we can evaluate phase quality by leveraging the occurrence of trios in MGI. So trios is like mom, dad, and child. And so, I mean, theoretically, at many sites, you should be able to look at the genotypes of a trio and then figure out the known phase of the trio child, and then you can compare that known phase to the statistically inferred phase to get an idea of switch error rate, or how often, is the phase incorrect. That is to say that the maternal and paternal haplotypes are switched between the trio child and its inferred phase counterpart. So we can use this to sort of inform on how well our phasing is doing, and then we can communicate this to investigators that are using the data, how well is it phased, and even within specific ancestries, like how well is phasing performing for different ancestry groups within MGI. Next slide, please. So I mean, as Matt mentioned earlier, kind of the landscape of technology is always changing in this field. He mentioned in particular the arrays, but also the reference panels that are available for genotype imputation are changing over time. And so a good example of this is a few years ago, 2017, 2018, we were performing our genotype imputation largely with the HRC, the Haplotype Reference Consortium panel, which is about 60K reference haplotypes, about 40 million variants, and it's predominantly European. At the time, this was one of the best options to perform genotype imputation, particularly in the context of reference haplotype number. But since then, the Transomics for Precision Medicine reference panel has become available within the last couple of years, which is more dense in terms of both haplotypes and variants, and then has the extra advantage of being comprised of haplotypes from different ancestry backgrounds, not just predominantly European. So the last two freezes, 3 and 4 from 2020, 2021, were imputed with both HRC and TopMed. Next slide, please. So TopMed imputed MGI is now the default analytic data set that we offer to users, even though we also offer HRC alongside. And the reason for this is just because it's more haplotype and marker dense. We can provide higher imputation quality and larger number of well-imputed variants, particularly in the lower minor allele frequency range of about 0.05 and lower, which is what this plot on the left is showing. TopMed offers quite a larger number of well-imputed variants for investigators to analyze. Next slide, please. So similar to how we have a metric to evaluate phasing quality, we also have a metric that we rely on to evaluate imputation quality, which is shown in the left-hand plot here. Empirical R squared on the y-axis, and then minor allele frequency on the x-axis. So empirical R squared is a metric that reports an imputation quality in ranges from 0 to 1, with values closer to 1, indicating the highest imputation quality. This plot is showing empirical R squared between TopMed and HRC. And so TopMed provides, as expected, greater imputation quality, particularly in the rarest minor allele frequency bins, which is one of the reasons why it's, our default data set to offer to investigators. And on the right-hand side, these 4 kind of tiny panels are basically the same thing, empirical R squared, but summarized across different ancestry subsets of MGI, so European, African, East Asian, and Native American. And in particular, we see a large boost in imputation quality for European and African subsets of MGI. And this is a reflection of the TopMed panel components, which are, there's a lot of European African components to TopMed. Next slide, please. And so, similar to what we discussed earlier with the negative control concept, there's also this positive control concept. Is there signal where we expect to see it? And so there's an image on the slide. What it's showing is we can take MGI genotypes and MGI phenotypes and perform association studies and look at those results, and also do the corresponding GWAS with UK Biobank phenotypes and genotypes. And what we would expect to see is where there's strong signal in UK Biobank, we would expect high replication in MGI, which is what this plot is showing. As signal increases in UK Biobank, we have a higher replication rate of that signal in MGI, indicating that not only is our genotype data likely reliable, but also our phenotype data is likely reliable as well. Next slide, please. So this is the last slide that I'm going to show for my portion, and it stresses this idea of transparency through documentation. So it's a lot of work to do the genotype QC and the phasing and imputation, and so we try and meticulously record how we do this process and then make it available on the MGI website, which is what that link provides. Is it links to both a list of the genetic data types that we offer, and then the accompanying documentation. And so these documentation pieces kind of serve 2 points. One is that investigators that are working with the data, it helps provide a detailed description of how that data was even generated, so that they can improve their understanding of the data itself. And also for our own internal work on the MGI side, when we do a data freeze, we can look at the previous set of documentation, the metrics that are summarized in there, and try and understand, are we increasing or decreasing our performance in certain metrics, and why or why not. So with that, I'll hand it back to Matt.
All right. Any questions? Anything so far? All right, then I will charge along here on the last. We're on our last section of this presentation. I'll try to get you to lunch on time. All right. So this is then talking about once we have believe that we have good data, both genetic data, clinical data, that's good, that they're properly linked. How do we integrate that data, and then how do we provide access to it in a secure manner? So data integration and data access. So, there are a very diverse group of people at U of M wanting to use MGI, right? And so they have a range of scientific questions and study designs, also very different in terms of their experience. There are people who are, highly trained at genetic analysis, and then what I'd call like recreational geneticists, right? People are sort of trying to take a stab at it, right? Using MGI data. And then there's also this large variation in their statistical skill set, right? I mean, how much do you know about these different tests that are being done, and then the computational resources, right? Do you actually have the computing space to be able to store the data set of this size, the ability to run a GWAS on 50 million genetic variants? Okay, so there's a range of people. We also have a range of clinical expertise, right? So sometimes, I might pick on people who are trying to do genetics that don't know the genetics and that well, but we have the same problem, right? People who don't understand the clinical part enough, right? And so we have to be able to work with them as well. And then we've got a range of data access, right? So specifically, if you are a clinician, right, and you have access to patient electronic health records, identifiable data, right? How do you deal with that issue? Okay. And so the ultimate goal of what we're trying to do in our group is get this data that we've spent a lot of time making sure that it's good, get it to the people that need it, and then get them the support they need so that they can answer their question, right? So people within school public health, the medical school, engineering, and now as MGI gets more publicized, right, this is expanding to people in other areas of the university. And again, you can imagine their questions and their expertise differ. The basic pipeline or thought process on here in designing a study is first you would start with the overall Michigan medicine patient population. You'd have to identify a cohort of people that you're interested in to being in your study. So this might be based on scanning the electronic health records, might be based on a chart review, right? So a lot of times clinicians who have access to more detailed clinical information, they might come to us with the patients that they've already identified. Sometimes it's their own patients if they have a specialty clinic. Sometimes these are genotype based, right? If you're interested in carriers of a specific SNP allele, you might do your study based on that. Sometimes people do it on all available samples, right? So once you have your cohort, then you need to decide on what kind of genetic data do you need, what kind of clinical data do you need, and then how do we place that data in a space that the user can securely and conveniently access it? Well, outside of our area, there's a group called DataDirect, which handles more of the clinical aspect or the clinical data aspect, right? So they have a self-service software tool for DataDirect, right? And so what you're able to do is you're actually able to query, perform this first part here where you identify your cohort, okay, from the full Michigan medicine patient population. One thing I'll ask you to notice here is that there's actually this level 1 login on this page. And so I'll come back to that in a minute when we think about safeguards for data. In terms of the genetic data, right? So, okay, so they'll handle the clinical end, all right? What about the genetic data? So we offer multiple versions of the genetic data set. So as Brett was mentioning, we have 2 different imputation versions. We do some filtering, basically what we think, if we were to run an analysis, here's what we would use. We also run a version that has no filtering done on it, right? Because, your question might not require that filtering, right? Especially if it's something like a minor allele frequency filter. If you want the rarest things, if we remove them, that's not helping you. We could offer just the chip genotypes. Sometimes we do custom data pulls where we phase haplotypes or just pull out, create smaller data sets that have just a specific set of SNPs and make it easier for people to work with. And sometimes we'll create entirely new types of data based on our existing data set to help out with whatever the user wants. And so I'll talk in a second about pharmacogenomic star alleles. So really, it depends on the user's expertise and scientific needs. And so we actually, when somebody initiates a request for MGI data, there's a consultation meeting that happens between a scientific research facilitator that's through Precision Health, a statistical geneticist from our group, and we discuss the project with the principal investigator and just try to understand what their goals are, try to help them with what the requirements would be to pull off this kind of analysis, and honestly also to talk a little bit about expectations on what they might be able to expect given the data that's available to them. Again, this goes into this expertise and people maybe who don't have familiarity working with genetic data. If you have 50 samples that are well genotyped, that might be really good for some studies, probably not great for a GWAS, right? So this idea of making data from data. So we have talked with people who are interested in star alleles, which are different haplotype allele combinations and a very specific set of genes that are related to pharmacogenomics and deal with how drugs are metabolized in the body. We've also worked with people who are interested in the HLA alleles, right? It's a very complicated haplotype structure within HLA. And so we've done some of the work to prepare for those. So briefly, within the star alleles, we worked with Amy Pasternak and Dan Hertz, who are in pharmacology, and they were creating data for an MGI, which actually wasn't as good, right? They didn't care about the SNPs. They wanted a very different type of data, these star alleles. And so we worked with them to understand what they were trying to do. And then we used our skill set to create better calls for these star alleles using some available software called Stargazer. And then they actually had some gold standard calls on some of these people. So we were actually able to look at concordance rates. So you could see in the people that we were able to do comparisons, it was quite high. Okay, so when we need to make new data, right, trying to help people out. So there's also people that don't have the bandwidth or the skill set to be able to do a genetic analysis themselves. So within our group, we've developed tools that overcome these lack of computational skills or resources, so that anybody who wants to run a genetic analysis can do it, right? It also avoids unnecessary data handoffs, right? When we first started doing this, we were sending people VCF files sometimes, and they would try to open them with Excel, right? And their laptop would start smoking. So two of the tools we've created are PheWeb and Encore. So I'll be honest now that I know Peter Vanderwerff is on this call. I feel like I shouldn't even be presenting this, but Peter was one of the main people that developed PheWeb. But PheWeb is a tool that allows us to interactively search all the GWAS that have been run on MGI, so something like 1,700 traits. And so Peter did an awesome job there, being able to do this sort of very interactive search.
Thanks.
Encore is another tool that's been developed. So PheWeb requires on pre-running the jobs. Encore is a tool that will basically, once you define your phenotype, will automate that parallel process to run the GWAS for you. And so it's got this really nice feature where you can drag in these phenotype files, and then that's it. You just hit go, and then all the magic happens on the back end, and then results are delivered to people. Right, so you can imagine how for some investigators, these are really nice tools to be able to not have to deal with some of the aspects that they aren't quite ready to tackle, but can still get their questions answered. So the last thing we're going to talk here is about data safeguards. So a lot of efforts put into creating this data, getting into the hands of users. The most important thing is that we don't want to jeopardize protected health information for the people that have consented to be part of this study. So basically what we mean here is that given health data, you shouldn't be able to determine who the patient is within the Michigan Medicine Health System. And so I'll mention three layers of data safeguards that we use within MGI. So there's administrative safeguards, technical safeguards, and physical safeguards, and these come from guidelines on security standards from CMS. So the first is an administrative safeguard. And so the idea here is basically limiting access to data. It's ensuring that only people who are properly trained and screened are getting access to this data. Okay, so when I mentioned that cohort discovery tool on DataDirect, there was that login piece, right? And so in order just to be able to do that, you need to have a level 1 password, right? So yeah, you need to be a part of the university. You need to have enrollment and duo authentication, right? So we need to ensure that the password has not been compromised. You need to have completed a HIPAA training, okay? So understanding the sensitivity of this data that you're about to access. And then having faculty status or faculty sponsor access for people within a group, right? So ensuring that the people who are accessing these data are people that are potentially are going to use them for research purposes. If you actually want to receive data, you need to fill out an IRB form. So that would be reviewed. That needs to be approved before you can actually receive data. Again, limiting access to people who have a legitimate scientific use for this data. Shifting dates of service, right? So that you don't really need to know specific dates usually, right? So doing that, you can still get at the longitudinal aspect. And then also providing access to high performance security storage at no cost, right? So if people don't have it, you can ensure that they have that secure storage. A technical safeguard is using technology to protect data. So a real great example of this is something like medical record numbers or dates of appointments or procedures are considered protected health information because you can figure out whose data that is. But usually research can be done without these things. And so we strip these things away from the data before giving them off to people in order to, as a technical safeguard, in order to protect the identity of patients. So here you can imagine when we have medical records on one side and genetic data on the other side, and we need to link these things, there's just no reason to give somebody that either these biobank numbers, which are BBIDs, or MRNs or medical record numbers. We just make up a new encrypted ID, right? And so we can share this with people. It links together these two data sets. People can get the answers they need, but we haven't compromised patient information. And the final piece is a physical safeguard. And so, this is that is trying to minimize unauthorized intrusion to this data. So the precision health computing environment. So there are both ARMIS or Linux and Windows environments that can both access where the data is stored on something called Turbo. The data sets are placed in Turbo, which is a HIPAA compliant computing environment. And the standard analytic packages are loaded there that most people need. And then you can work with your research facilitator to get new packages if you need them placed there for you. And there's limitations on importing and exporting files, right, to prevent people from taking this data out of this secure environment. So that's an example of a physical safeguard that gets put on this. So I'll end here just by saying, this is a lot of work to go into creating this data set, to maintaining it, the rigor that goes into making sure that it's of good quality, and then the steps that need to be taken to then distribute this data to the university. So why would you create this data set? Like what's the value of it? So we're just starting to see now, U of M, what can come out of this, right? And so it's this idea of an accelerator and an incubator for interdisciplinary health research. So I just want to mention this study that I was a part of and I helped with. So Carolyn Swenson is an MD in the Department of Obstetrics and Gynecology. So she actually left. She went to Utah, which is very sad. She was a cool person. I liked her. But her work focused on improving health outcomes in gynecology, and so particularly preventment and treatment of pelvic organ prolapse. She was not a geneticist at all, had never done a genetics project, but she was interested in asking if there was a genetic basis to some of these conditions that she was looking at in the clinic. And there was not much in the literature at that time. So what would it take for somebody like Dr. Swenson to do a GWAS of the work or of the phenotype that she's interested in? So first, she would have to identify and recruit cases and controls, right? So that takes time if you don't already have them, or you've got to recontact them if you do have a list of people. You'd have to get blood samples from these people, so there'd have to be a physical interaction with them. You'd have to have the expertise to be able to do DNA extraction. You'd have to store them, biospecimen storage. You'd need to pay for genotyping arrays, the cost to do the processing. Then you'd need to have a place to store that data. You'd need to have high performance computing access to run the GWAS, and you potentially might need to have a statistical geneticist to help out. So for somebody like Dr. Swenson, who is primarily a clinician, right, didn't have these things. So what would that mean? It would mean she needs a grant, right? There's the amount of time and the money it would take to pull off an analysis like this. Well, alternatively, because a resource like MGI exists, we met with Dr. Swenson in one of those consultation meetings. She had the clinical expertise. She was very clear, like this is what I'm looking for. So she was able to come up with a definition based on these ICD codes. She worked with DataDirect to pull out the people that she wanted to be her cases and controls. We worked with her on doing the genetic analysis on our end. And I'm happy to say that just very recently, she had a paper accepted for a genome-wide association study of pelvic organ prolapse using the Michigan Genomics Initiative. So the data set wasn't massive, or this case size wasn't massive. We didn't identify anything novel, but we actually replicated 4 biologically plausible SNPs. We did some comparison to UK Biobank results. And I think actually one of the most interesting things that came out of this is that we debunked some previously reported SNPs that were very, very likely to be the result of genotyping artifacts, and we were able to show where those genotyping artifacts might have come in. The power of MGI then is we can just reload this thing and take the next trait. The overhead has already been done. The QC work has already been done. The next GWAS is already ready to be run quickly and inexpensively. So, it doesn't matter what that trait is, right? This data set can be used for it. So I want to thank the people that have been involved with the Precision Health Initiative and have worked on this data across many different departments. And I will stop by leaving this list, which I always like to leave here, the top 10 most hilarious ICD codes as pointed out by this site. But happy to take any questions.